All right, so I'll be starting on a series of chemical pathology questions today, and I hope to do 40 videos today. All right, because I have a total of about 120 questions, and I would like to be doing 40 every day. Okay, so in 3 days I should be done doing the chemical pathology questions. Okay, so in these questions, let's look at the exam question. The students in an exam were asked to describe an apolipoprotein, and this was using a schematic if possible. Now, if they ask you to use a schematic if possible, they are not asking for your opinion. You must use a schema. And what is a schema? Schema is the drawing like this. Okay. So here, yeah, we can't draw, but at least I brought this image so that you guys can see what an apolipoprotein looks like. So an apolipoprotein is not really a single molecule, it's a part of a lipoprotein. Do you understand? So for you to actually draw an apolipoprotein, you have to actually draw the lipoprotein. Do you understand? Then include the apolipoproteins in it. So from here you could see what apolipoprotein A, the lipoprotein B, the B are three to show that there are more. Okay. Then we have the apolipoprotein C and the apolipoprotein A. Okay. So this is how the drawing looks like. Okay. But I think let's just talk a bit about the apolipoprotein, because they ask you to draw, you don't just draw only, you have to at least give one or two paragraphs about it. So this apolipoproteins, we say that they are structural what constituents of lipoprotein particles. Okay, so they are okay, these structural constituents of lipoprotein particles are polypeptides that are involved in metabolism of lipoproteins. Okay, and when you are talking about metabolism, you are talking about anabolism and catabolism. Okay, and anabolism is like the build up. So anabolism involves what secretion and processing. All right, then catabolism too. So they are actually constituents of lipoproteins. Do you understand? But they actually take part in the metabolism of lipoproteins. Okay, so talking about their classification, we say that they are classified what alphabetically. That's why I have apolipoprotein A, apolipoprotein B, apolipoprotein C, and apolipoprotein A. Okay, we say that they are also subgrouped into the non-Roman numerals. Okay, so coming back to these headings, these apolipoproteins here, we were to describe them, describe the structure and the functions. Sorry, I forgot to put functions here. And functions is what we're going up next. So these are their functions, but I feel like let's talk about it from this point of view first. So one of their functions is what? For transportation. Okay, so apolipoprotein A is for what? Cholesterol transport, while apolipoprotein B is for what? Triglyceride transport. Okay, and another function they subserve is what? To stabilize micelles lipid structures. Okay, they stabilize micelles lipid structures. Okay, they can also act as what? Enzyme cofactors, like they are cofactors to enzymes. They can be activators, they can be inhibitors. That's why I see along all these processes, you see them being active along the process of what? Lipoprotein metabolism. Okay, so they can act as enzyme cofactors, activators, and inhibitors. They mediate the binding of what? Lipoprotein particles to receptors in the liver and peripheral tissue. Okay, so they mediate the binding of these lipoprotein particles to receptors in the liver and the peripheral tissue. Okay, so now the ratio of apolipoprotein A to apolipoprotein B may be useful, may be a useful measurement of what? Cardiovascular risks. Okay, so now that's if the ratio is more than one. Okay, so now for the ratio to be more than one, that means apolipoprotein A is more than apolipoprotein B, which is not supposed to. All right, so looking at this image, ideally apolipoprotein B is in excess. Okay, apolipoprotein B is more than the other apolipoproteins that we have. Okay, so now this ratio is useful measuring cardiovascular risks, and it is significantly influenced by the status or by the fasting status of the patient. Okay, so you could see apolipoproteins. This is just a schematic trying to show you guys how they are involved in the various metabolisms of lipoproteins, okay? But the best one for you to actually go up with if you want to draw this is this, okay? It's this, all right? It's this. Now you can see this. These ones are what? Cholesterol esters, all right? So you might want to actually do a point out here to put out the cholesterol esters, okay? Then the ones inside are what? The ones inside are the what? Triacylglycerol, okay? So cholesterol esters, triacylglycerols, okay? So that's it about what? Apolipoproteins, their structures and functions, okay? So bye.